So first of all, thank you very much, all of you, for joining me in this evening, and particularly to the ninth lecture in the ninth session in the BioEdge series, which is helping you to create a small edge over through the learning from Biocon Academy. 5 minutes, I mean, 5 or 10 minutes probably. I was taking a coffee before this session. When I was taking the coffee, I was wondering how it would have been 300 years before. Uh, 300 years before means we are talking about pretty much the early stages of the discovery of coffee, which means the coffee beans are harvested from the field, and it is grounded manually, and it's heated up, and then it is grounded manually, and everything is manual. Completely, it is a manual procedure. From manual procedure, what is the first change that has happened? What is the first revolution that has happened? By the end of this slide, I would like to ask all of us a question, whether we should call this as a revolution or something else. The first revolution is that whatever human being, or whatever the manpower, we were doing through our hands. For example, plugging off the leaf, or taking away the kernels, coffee kernels from the plant, heating them up, and grinding them up, and removing the impurities, and so on and so forth. Whatever manual work we have been doing, or even packing, whatever manual work we have been doing, 1780 witnessed the first industrial revolution, where most of the things moved into the mechanization. Everything has become mechanical, which means that most of the things, whatever we are doing by hand, the machine started doing things. It could be this machine could be run by heat, or by steam, the heat that is produced by steam, and so on. Which means it is a clear replacement of the human efforts by machine. And what is the greatest advantage? What is the greatest benefit? What is the greatest revolution we saw at that time is that it's a 40 to 50 times efficiency increase. How many grains, how many coffee beans one person can plug and grind? It is very different when compared to how much a machine can do, which means the machine started giving 40 to 50% more efficiency when compared to the same thing that is being done by human hands. That was a huge revolution by converting the human efforts to machine efforts. Then came electricity. When the electricity came, everything became electrified. It could be the train, it could be the machines. The steam machines are converted to electric machines. The coal-based trains are converted to steam-based trains, and then to electric trains, and everything has become completely electrified. This electrification has created a huge revolution in the way we looked at the entire production or industrialization. It is not only the industrialization, in our regular life also, in our day-to-day life also, this electrification brought up a lot of changes. The way the education is looked into and all that. If you carefully see, between 1780 and 1870, we are talking about approximately about 90, 100 years. So it took about 100 years for a new thing to come in and revolutionize. When you take the example of the next revolution between 1870 and 1970, the same, pretty much the same time span taken from moving from the electrification, mechanization to automation. Because automation is not very simple, because there is a HMI, there is a human machine interface. This human machine interface is very, very important to convert what human think the machine has to do and the machine has to do, which means press of a button, press of a touch screen, and so on and so forth. In this 100 years, this complete automation has become a huge revolution. Up to here, I'll stop here, and then we'll think for a second. When the human efforts converted to mechanics, from mechanics when it is converted to electrification, from electrified instruments when it is becoming automated instruments. What happened to the talent? What happened to the human power, human resource? Has it shrunk? Has more and more people become jobless? The surprising answer is absolutely no. The industry became mature. When the industry became mature, the people who are working in the industry also matured along the way. They picked up the skills that are required to be in that industry, and the talent pool and the manpower continued to grow, which means that the demand for the manpower is always there in spite of the mechanization, electrification, or automation. No exception for digitalization. I'm sure, though it has started in 2011, 2019-20, we absolutely witnessed this digitalization in all forms, particularly the money. If you remember, in 1990 or 1995, if you want to remove, or if you want to withdraw money from your own account, you have to go and stand in a queue in a bank, right? Or if you want to transfer money, we used to issue check with the hope that the check will be honored. We will receive the check with the hope that it will be honored. These are not the case now. It is all digital money, digital economy, completely digitalized. Not only money, you can take any sector. It could be entertainment, it could be leisure, it could be travel, anything you take, everything has become digitalized. This is an era of digitalization. But if you want to carefully see the time taken for digitalization from automation, it's about 40 years. From 40 years, because we have such a strong backup of revolution over revolution happening over 300 years, now we took only 9 years to move from digitalization to personalization and humanization. Now everybody thinks that the machine should support the way I want. The machine should support the requirement of mine, my requirement. I'll give you a very, very simple example. We used to get a toothpaste advertisement, which is called a Chyawanprash toothpaste, for example, Dabur Chyawanprash. This Dabur Chyawanprash is a toothpaste advertisement for everybody. It is for the mass, which is not addressing those people who do not like that smell. It is not addressing to those people, those who do not like the color of the paste. Now, we have come to a stage where this digitalization has created digital data pertaining to each person, which is able to personalize things for each one of us. Which means if 1% of the population is actually liking white color toothpaste, now we are able to advertise white color toothpaste for that segment. If somebody, I saw a advertisement, I don't remember the brand name now. There is a purple color toothpaste, blue color toothpaste. We would have never imagined using a blue color toothpaste, a complete blue color or purple color toothpaste, because they say that it is good. It removes the yellow coloration of the teeth. So it is all about humanization and personalization era. The reason why I wanted to spend this 5 minutes in industrial revolution before we jump into artificial intelligence in life science is that to clear out the air, to clear out the fear that these technologies will not take away human jobs. More so, it is going to help increasing the demand for a specialized manpower, which means that, as I already told you, the human being coped up in each stage. When I say coped up, they skilled, they upskilled, they develop skill, which is required for that particular technological era. So it's true for the industry 5.0 or 4.0 currently running. So we are now talking about artificial intelligence, machine learning, and deep learning. That does not mean these machines learn the way we work and replace our work. Even today, these are in our control, which means we need people who has this background, who has this understanding to run the show. With this positive note, I would like to enter into this session 9, which is artificial intelligence in life science. Before we get into details of what are the tools that are used in life science, what is the term use case actually mean and all that, we'll set the platform clear. First, we'll define what is the scope of this presentation. This presentation, we are going to restrict our discussion on application of artificial intelligence in the life science area. Life science area covers three things: pharma, biopharma, medical device. These are the three things the life science area covers. When I say pharma, we are talking about small molecules, the biotech, large molecules, medical device, you know very well. If you were a part of the earlier BioEdge series, we spent a lot of time in deliberating on how you should understand the small molecule and large molecule. That is the reason why I'm not spending much time in that. I'm directly considering these as pharma products and biotech products based on the origin, the chemically synthesized molecules and biologically synthesized molecules, and those instruments and aids that are used in the diagnosis and treatment and prognosis, monitoring the disease management in the life science area, that are medical devices. So this is the scope in which we are going to speak. We are going to speak about applications of AI in pharma, applications of AI in biotech industry, application of AI in medical devices. This is how it is structured for today. Before we get into some of the examples and basic principles behind or the basic architecture of the artificial intelligence, I would like to spend little time in how it is moving, in what direction it is moving. It all started with AI. All of you will agree with me. AI is not a futuristic technology. We will be ridiculed if we say artificial intelligence is a futuristic technology. It is a current technology. In fact, it is not even current technology. I won't mind, I won't dare, I would dare to argue that this technology is a little old technology. Artificial intelligence is 1995 technology. In 1995, in a conference, it's just happened as an artificial intelligence to understand how to make a machine think like a human being. Then we started moving into generative AI. In generative AI, it is a very fascinating work we have done because we have created algorithms that can create the result a human being can create without the guide and support of human being. For example, I'll take an example of what is the name of the movie in Kannada? I don't remember now. Love You, if I'm not wrong. Those who are from Bangalore who are listening to me right now, you can put it in the comment if I'm right in the name of the movie. Love You, if I'm not wrong. It is a 100% AI movie. It's a feature film created by generative AI, which means not even one human character in it, but it is a full featured movie. It is not an animated movie. It is a Gen AI movie. Okay, generative AI movie. Gen AI need not be that complex also. It can be very simple also because it is more about content generation. The content could be digital, the content could be written, or content could be anything. It could be even a text, for example. For example, let's say you are asked to write an assignment. You are asked to write an assignment of today's learning. It's a very simple thing. You can just go to Ask Gemini or Copilot or Perplexity or what do you call Chat GPT, these tools, and just ask them to produce a result for you. It is absolutely available for you. Okay, it can generate a report as if you have generated a report. It is generating on your behalf. Now, I would like to take one step behind and think, how can a computer think the way you think? It is not creating a generic report. If you carefully see, it is not a generic report. It is pretty much aligned to the way you would have written it. which means it understands the pattern. It understands how you work, how you speak, how you write. It absolutely understands that. Okay. There are so many examples you can take for this claim that the machines are actually listening to you. You sit in your living room, you speak about a blue checked shirt with your family members and express the interest that I would certainly like to buy a blue check shirt. And the Siri or an Alexa sitting there in the living room understands this. Next time when you open up Instagram, next time when you open up your Facebook, there is an advertisement of blue check shirt. So absolutely everything is customized for you. This is the reason why I'm telling you now you are getting into humanization. Now you are getting into customization, personalization. It is a very high level understanding. We'll get into a little more deeper understanding moving forward. The AGI, that is the general intelligence, artificial general intelligence. Artificial general intelligence is a sum total of everybody's intelligence put together, which means it is slightly above the way the human can think, which means it can create certain things the human wouldn't have done otherwise. We have not yet started working on AGI based platform very frequently, but it is certainly in use. The artificial general intelligence is having multiple applications in different types of industries. Then it is artificial superintelligence. Artificial superintelligence is going to create certain things which human being cannot create. When I say cannot create, the volume. For example, if suddenly you want 5 million data points, synthetic data points, you will not able to create in a shortest period of time, where ASI can. So the artificial superintelligence is going to be super intelligent than AGI, which can create data sets or can do certain things much more than the human being's normal efficiency span. The last one is much more interesting. Imagine what if you can have a small brain outside of your body, outside of the control of your central nervous system. Or if you are a biotechnologist who is listening to this session, what if you can culture your pluripotent brain cell on a chip? This itself is called as brain on a chip. It is called as organoid intelligence. In organoid intelligence, you can create organoids, which organoids are nothing but the artificial organs, organoids. And the term intelligence is added to this because organoids are nothing new to the industry. We have been creating electronic and medico electronic arms. Absolutely, there's no problem. But only difference is that these arms, these organoids cannot think by itself. But the organoid intelligence can think for itself. That is going to be the future. Organoid intelligence. I'll spend little time in organoid intelligence in my last slide. To we already spoke about what are the life science segments we are going to talk about. What is the trajectory of artificial intelligence in an overall sense? Please remember, all these can be called as artificial intelligence, whether it is OI or whether it is ASI or AGI. Everything can be called as artificial intelligence because artificial intelligence is considered as an umbrella term. Everything can be put under the umbrella of artificial intelligence that we have seen. Now, I would like to spend few minutes in rehearsing or bringing up to your understanding on what are the common platforms on which these artificial intelligence algorithms work. One of the common is called as KNN, otherwise called as K-near network, neural network. K-near neural network is a very simple way of working. In the questionnaire, I saw some of the questions like how artificial intelligence can help a microbiologist. If you're a microbiologist who is listening to the session, you might know that there is a concept called as API, that is Analytical Profile Index. In Analytical Profile Index, you are creating a code for the microorganism based on their biochemical reaction. If I take an example of IMVIC, IMVC, indole methyl red Vogus Prosker citrate utilization test, IMVIC test, you have plus plus minus minus, minus minus plus plus, which means plus plus minus minus is equal to E. coli. Minus minus plus plus is equal to Enterobacter cloacae. This is the code. What if this code can be understood by a system? And whenever the system is going to see this particular code in any of the document, it understands you are referring to E. coli, you are referring to Enterobacter cloacae, which means you have a constant and you have a test instances. In the testing instances, it is going to find out wherever the constant instances are overlapping. Based on that, it understands what you're talking about. This is called as K-near neural network. The FFN is very easy one, which is called as forward flow neural network. In forward flow neural network, you have an input layer, you have a hidden layer. The hidden layer is called as a processing layer, and you have output layer. The output layer tells you what it is. For example, in the input layer, if you take a pattern of your search in the Google search, the Google search terms is your input layer. The system, this algorithm, if it is operating in your computer, this computer is going to learn, it is going to watch what are all the things you are searching in A. Are you searching Agra? Are you searching algebra? Are you searching adult? Are you searching whatever? Okay, in A. For example, these are the three things you have searched always. Always you have searched adult, you have searched algebra, you searched Agra. These are the three things most of the time you searched. These three are called as input layers. The hidden layer prioritize how many times you searched adult, how many times you searched Agra, how many times you searched algebra. In the output layer, when you start typing A in your Google search, the first one is adult. How it is adult? Why not algebra? Why not Agra? It is because the hidden layer processes saying that this is your top priority. That is how the forward flow neural network will work. It's a very simple algorithm which is used for autofilling. It is not only used for autofilling, by the way, but one example I am taking is autofill. The next one is a slightly complex one, where you can imagine here is also there is an input layer, there is an output layer. Only difference is that.
This is pattern recognition.
Hello.
Exactly.
Can you
Sorry,
please
sir.
mute the line? Okay. So in the convolution neural network also, there is an input layer, there is a processing layer, and there is an output layer. Only difference is that the processing layer is very, very complex. That is the reason why it is coming under DL. That is a deep learning network. The difference between artificial intelligence, machine learning, and deep learning is that in artificial intelligence, the machine start thinking the way the human being think based on the pattern. In machine learning, the machine understands the pattern and make the prediction. In deep learning, the prediction is validated and accuracy is improved. This is how you can simply keep the understanding between AI, ML, and DL, examples being KNN, FNN, and CNN. Where is the application starts? There are so many ways this AI, ML, DL is applied. For this presentation purpose, we are going to focus on NIP, natural image processing. Natural image processing and natural language processing cuts across different things, different aspects of artificial intelligence, machine learning, and deep learning, because all are required. I'll take an example for you before we get into the actual technical examples. This is called as a computer vision. When I say computer vision, there are different ways you can understand this computer vision. For instance, you have a laptop and you have a computer vision algorithm. You can show a cat in front of the computer and ask the computer to recognize what animal it is. Pretty much possible. It is quite possible. Or this computer vision can be a small part of integrated computer network in an overall robot. For example, there are two eyes for a humanoid robot. These two eyes are linked to the algorithms for the computer vision. This computer vision is working on the natural image processing algorithms. Now, how it works. It doesn't take much time. It takes fractions, millifractions of a second. Initially, what it does is it completely depixelates it. The way exactly the other way around of the human brain works. Human brain works from pixelation. The computer vision works from depixelation. It is completely depixelated to individual pixel level. After the pixels are created, each pixel is given a number. This number is the RGB number. All of you are familiar with the RGB. There is a red, green, blue index, RGB index. And this RGB index number is assigned on two levels. What is the color and what is the density? So based on the color and density, these numbers are given and it is distributed. Now, only thing the computer has to do is it has to do a little bit of this sounds absolutely ridiculous, correct? I'll tell you why I specifically use the term ridiculous because if it is making a mistake in identifying a cat as 82% cat and 15% dog, it's perfectly fine. It's okay. Nothing happens, right? As long as it is not for a very critical operation. If you're just taking a trial on computer vision, it doesn't make a big difference. It's only about enhancing the algorithm, fine tuning the algorithm. Only that much it takes. But what if it is used for more critical operations? This slide I would like you to remember carefully because this is the way the computer is going to use or run the AI algorithm for life science applications. As I told you, I take an example of k-near neighbor factor. In k-near neighbor, what is the nearest neighboring factor or data point to the constant? As I already told you, the constant is nothing but the structure or the pattern the computer has created based on the machine learning from the so many number of cat pictures you have taken, for example, or so many pictures of cat you have trained the system with. Now, when you show a cat, it is the input layer. This input layer, now it is going to take a picture or take a look at it and trying to understand where it is fitting based on this vector analysis. I'm not going into the max part of it. We will go into directly the application. One by one, we will see the application. The first application we would see is AI in pharma and biopharma. I really would like to thank those who have specifically shown interest in the specific area which you want to listen to. This helped me putting this slide because otherwise this particular slide was not there in my deck today morning. After when I spoke to Nabil, he was sharing the list of things as a expectation from this session. Super helpful. Thank you so much. One of the question was how to use artificial intelligence in drug discovery and development. All of you may know drug discovery and development is a very, very long process. When I say long process, it takes at least 14 years to discover and develop and market a drug if it is a new drug in a conventional way. With use of bioinformatics, with use of artificial intelligence, the cycle can shrink a little bit. or if it is a completely a different pathway, like if it is a biosimilar. If you remember one of the sessions where we spoke about different types of product lines in the pharma biopharma, we spoke about biosimilars. So if it is a biosimilar, there is no need for an extensive developmental pathway. That is, phase one, phase two, phase three clinical trial need not be done so extensively. There can be possibility that it can be waived off because the data is available for the existing molecule, that is a RLD, reference listed drug or a patented molecule. Now, this could be an excellent use case. The term use case is very important, which means you are not applying the artificial intelligence algorithm in random things yet. You are choosing a situation, you are searching and you are choosing an instance particularly for you to best use the AI ML DL. One of that is drug discovery and development because of the time it is taking. It takes time because, as I told you, in the one of the sessions we went through in the BioEdge series itself, in the earlier stages, the discovery itself has a lot of things to do, starting all the way from target validation, target identification and validation, hit identification, that is the therapeutic molecule identification, lead optimization, lead identification, that is whether the target and the ligand are binding to each other with strong chemical affinity or not. Then lead optimization, which is chemo, uh, a little bit of chemical work and omics work to increase the binding efficiency, that is lead optimization. Then candidate, when the candidate means it is converted to a drug product, what is the format in which it has to be tested, then it is tested on the preclinical uh, testing. In preclinical testing, it is tested on the animal models. After animal models, the data is collected, the data is submitted to the agency look into it, and then if you have done everything right and the agency feels it is safe to move forward, then they give the permission for clinical trials on human being. You have clinical trial phase one, phase two, phase three. I am not at all spending time in this because this we have done already in one of the session. It is just a repetition. The whole thing I wanted to talk to you here is the number. If you see the blue color numbers, this is the number of years it takes in each of the stage: discovery, development, and marketing, or bringing to the market. And there's the amount of money that is being spent in each of the states. Some of the money is hidden. For example, clinical trial itself takes about $400 million. So totally we are talking about anything between $800 million to $1 billion. The stake is really very high. Any amount of time you can bring down in any of the stages is going to be beneficial, both in terms of early reach to the market as well as the amount of money spent in each of this item. This brings this particular point to be a best point, or it's called as a use case. Now we will see one of the algorithms used in reducing the overall discovery time. If you want to discover a completely a new molecule using generative molecular design, this generative molecular design is nothing but based on the existing compounds. If you have a new target, I'm sure all of you understand the term target. Target is a proteinaceous substance, that is proteinaceous or protein carbohydrate combination found on the cell surface. It is called as a cell marker, receptor, biomarker, whatever you want to call it, right? So the target. So if you found a new target and you want to find a completely a new molecule for this target without doing complete research in the initial stage, you want to find out from the existing molecule which one can fit properly. For which, first you need a working compound library of various molecules. These various molecules are fed into a proprietary prediction pipeline for the machine learning. The machine learns through multi-level models and system models in terms of these parameters. It is not understanding the molecule just as a chemical molecule. For example, let's say paracetamol. If paracetamol is one of the molecule in the working compound library, it understands what is the efficacy of paracetamol as an antipyretic one. as anti-cancer, anti-fever treatment or anti-inflammatory. It is a NSAID, right? It is a non-steroidal anti-inflammatory drug. So as a NSAID, how effective it is, how safe it is, what are the side effects, what are the reported ADRs, adverse drug reactions, what is the PK profile, pharmacokinetic profile, what is the ADME profile of this particular paracetamol, how easy or difficult it is to develop as a molecule. Is it a very complex one? Is it a simple one? If any changes happens, will it lose its efficacy? How easy to develop? Based on these properties, it understands. These are called as tags, TAGS tags. From AI perspective, the molecules are tagged with this data. This data is called as a metadata. Metadata are nothing but data on the data or data about the data. Paracetamol is a data. Efficacy, safety, PK, developability are the metadata of paracetamol. Along with that, it understands paracetamol. Like that, thousands and thousands of molecule from the compound library, working compound library, it tries to understand. Now, you are going to give a design criteria. Here I will use the, oh my God, no. Here I'll use the pointer so that you can navigate it better. Yeah, here you are going to give the design criteria. The design criteria, what you want is, you want it for the particular target. The machine now has information about, for example, 20,000 compounds from the working compound library. From this 20,000 compound, you want a compound for a particular target which has a particular efficiency. When I say efficiency, bioavailability, pharmaceutical availability, so on and so forth, with a particular age group and with particular toxicity profile, which means safety profile. If you give this design now, based on your design, based on the learning, it is going to generate molecules. The system is going to generate molecules. The system, when it is generating molecule, it is generating in two levels. One level is simulation alone is sufficient to validate your data. Only the simulation studies, for example, it could be ligand binding, chemical binding studies, or QSAR, that is a quantitative structure activity relationship, is it sufficient? Or do you need a complete experiment to be conducted? In two levels, it will give you suggestions. It is up to you. For example, it is up to us to decide what type of decision you want to take. Whether you want to take a decision based on the simulation or you think experiment is needed, or both. It is up to us to decide. Based on that, you will do that, and the data will be generated for those molecules. Those molecules that are generated against your design criteria, you can do a simulation study, you can do a experimentation or human relevant assays and add the data to the generated data. And again, that goes back to working compound library for comparison with the existing molecules. Again comes back. When you come back again, now it is not 20,000 molecules. Now it is only 6 molecules or 7 molecules. Out of the 7 molecules, again you run this cycle. You will drill down to 2 or 3. Again you run it, you may drill down to 1. This one molecule has a probability of lot of success if you take it forward into your drug discovery. This is how the drug discovery cycle of 6 point years can be reduced significantly by using AI tool. For example, I'm talking about Atom for small molecules. This is one example. Next example, I'll take it from pharma side. Pharma biopharma application. In pharma biopharma application, there is a particular um, department called as quality control. In quality control, there is a test called as visual rejection. As I told you, in the past, everything was manual, then it became automated, then it became digital. Same thing happened for the visual reject also. In visual rejection also, earlier used to be, we will take the sample vial. After the vial is filled with the medicine, you take the vial and see it in against one white background, one black background, which is called as 4-eye method. Two with white, two with black. So 4-eye you are seeing and see if there are any suspended particles. This is a very laborious work. Then this was converted to a spectral method, which is a little more sophisticated one. It is a mechanical, it is a automated method, spectral method. Based on the digital data you are obtaining from the spectral method, you will be able to decide whether there is a crack or whether there is a suspended particle or if there is any deformity in the vial or not. Okay. Now, I would like you to imagine only one thing before we get into what is the role of artificial intelligence in visual inspection. There are two things which will create the same impact on the vial, in the glass vial. If the glass vial touches shoulder to shoulder with another vial when it was hot, it will create a weld mark. Weld mark means there is a mark that happens on the shoulder of the vial when it is touching each other under high temperatures. For example, deep aerogenation tunnel, it is at 350 degrees centigrade. So sometimes it may touch and go off. In those cases, it has created a weld mark. This is instance 1. Instance 2, it may not touch, but the vial itself, because of the manufacturing defect in the vial, it may have a small crack, which is not visually observable. Sometimes you will not able to observe it when it is there in the plant. If it is in your control before it is reaching the market, if you are able to identify the crack, it is well and good. What if it goes to the market, then the crack is identified, which means the product becomes turbid and then it comes as the return, as a recall. Then it is a huge problem. Now, the spectral methods cannot differentiate between a crack and a weld mark because it is just based on the wavelength study. It's a wavelength diffraction study. This diffraction study really cannot differentiate between these two. Now, if you start using this, or if you want to consider this instance, it is an excellent use case because each vial is very, very high cost. Each vial is very expensive. When I say vial, I'm talking about the content in the vial. For example, if the content in the vial is a monoclonal antibody, we are talking about thousands of rupees for each vial. And if I'm manufacturing 1 lakh vial, out of 1 lakh vial, if I get a visual reject of 5%, it's a huge one. It's a huge number, right? To be considered as a use case. Now, what people started doing is after you identify, after it comes back from the market as a crack or a weld mark, they start generating the data with photograph of weld mark and crack mark. A human eye cannot differentiate, but a computer vision can. Therefore, it is these photographs are trained are used to train the computer using a CNN platform where it continues to learn from the photograph and the tag whether it is a crack or it is a weld. The reason why it has to differentiate is because if it is a crack, it is a true positive, which means the vial shouldn't have reached the market. If it is a weld mark, it is a false positive, which means that the weld mark is not going to impact in any way or it is not going to interfere in any way the product quality. It can go into the market. Now, if I can differentiate between the weld mark and crack mark and recover at least 50% of the results from a visual rejection, that is an excellent use case. That is where the trained convolutional neural network is used. As you can see here, the trained convolutional neural network, and when it is deep neural network, it is moving to 16% to 83% of accuracy, from 21 to 64% of accuracy. When I say accuracy, which means that when 37 false ejects were applied to two different trained models, the models extracted good ones with the accuracy of 64%, which means 36% I am correct, which means that many vials are recovered. This is a huge cost saving from the quality control that can give back to the overall cost of the vial to the organization. Excellent use case from pharma biopharma from use of the NIP, as I told you, natural image processing, in the CNN algorithm platform. Is it new? Is this artificial intelligence or machine learning or deep learning, is it new or is it not being used by the industry? The answer is no. IBM Watson is one of the very commonly used platform by Pfizer for immuno-oncology drug and diagnosis market. Sanofi is using the artificial intelligence network to hunt for new therapeutic targets for metabolic diseases. Okay? The Roche is using the reverse engineering and forward simulation platform to analyze the cancer patients to identify new targets. Because more you identify new targets, more drugs can be manufactured, I mean, discovered and brought into the market. These predictions makes a huge business impact. The last but not the least, I'm going to take the example of medical device, sorry, medical device, which is used for diagnosis. This is where I would like to bring to the point. As I told you, whether the computer vision understands a cat to 82% or not is not a big deal, as long as it is not a critical usage. But in case of disease, for example, I am taking an example of breast cancer here. In case of breast cancer, if the diagnosis is early, if the breast cancer can be diagnosed early, if the breast cancer can be diagnosed accurately, reducing false positive, reducing false negative, this entire thing is going to increase the productivity and longevity to the patient. What do you mean by false positive? False positive means the patient is not suffering breast cancer. For whatever reason, the oncosurgeon or the oncophysician has come to a conclusion that the patient is suffering uh, breast cancer, is developing a breast cancer, for which the patient undergoes surgery, undergoes chemotherapy, radiotherapy, and they go off with that. Very, very painful, right? Should have been avoided or could have been avoided as long as there was a technology. False negative is all the more pathetic. False negative means there was a breast cancer. The diagnostic method is not able to give accurate data to the oncophysician and oncosurgeon to decide that it is a breast cancer. Therefore, it is missed, which means the patient continues to develop breast cancer and suffer because it is not identified in the early stage. Now, if I come back to the number, if I say the patient is having 82% breast cancer, what about the remaining 12% or 18%? It could be false positive, could be false negative. We really do not know. Or this 82%, if I question what is the accuracy? If it is 82% with 80% accuracy, still we are talking about 16, right? So it's a huge use case for diagnostics to use artificial intelligence, particularly the NIP, natural image processing. In natural image processing, I take example of convolutional neural network here because the classifying layer is very high here because we need multiple things to be analyzed at the same time. When I say multiple things, it could be the mammogram, it could be the what do you call the prescription written by the doctor, the mammogram with different contrast levels, or it could be the conversation between the doctor and the patient. It could be the prognosis monitoring report. All that are important. Everything is a metadata. All these are data around the data of that particular mammogram of that particular patient. Again, we'll go back to the vector analysis. In vector analysis, the same way as I told you, the CNN also does the same type of works based on the same vector and tries to understand the same area with the different contrast, different color allocation, which is not possible from a human eye perspective. I cannot naturally do it. Artificially, you can do it. In a system, you can do it, but more accurately can be done by the AI. Now, after this analysis, or after analysis, when you feed all this data into the computer, which is going to work on CNN platform, it will take all this as an input area or input layer. From this input layer, it can create multiple uh, hidden layer. When I say multiple hidden layer, each layer can analyze one parameter at a time. What is the age of the patient? What is the physiological pattern of the patient? What is the stage in which the patient has come to the doctor? What is the mammogram analysis say by the pathologist? Whether the pathologist report is correlating to the mammogram. Because after the identification in the mammogram, they would have taken a biopsy. Whether the biopsy result is correlating to the mammogram. All these are identified separately as a layer and then come to a conclusion. The machine learns. The machine learns more and more. As I already told you, you have to make the machine learn on the outcome also. Which mammogram has resulted as a true positive, which mammogram has resulted as a false positive or false negative based on the historical data. This is a very difficult stuff, but we have to do it. Only if you do this in future, you will be able to get better accuracy. With this type of diagnostic tool, you will be able to get higher accuracy in the way the diagnosis is done. I'm going to give you one predictive NIP video for you to watch for
With
just a few
advances
seconds.
in machine learning and artificial intelligence, a new role is emerging for machines as intelligent assistants to radiologists in their clinical workflows. But what systematic clinical thought process are these machines using? Are they similar enough to those of radiologists to be trusted as assistants? In this live demonstration, clinicians can select a case from various subspecialties, attempt to make a diagnosis, and see how a work-in-progress Watson technology attempts to assist the same case. Watson uses sophisticated medical imaging, deep learning, and clinical inference technologies to analyze patient cases using a systematic clinical thought process. To experience the eyes of Watson, you can select a case for analysis. You can examine the imaging study and read the associated case description. Select the appropriate conclusion. To see how Watson would attempt this case in real time, select Ask Watson. Watson examines the case description first, analyzes text, and highlights the relevant clinical concepts found. It summarizes the findings and updates the evolving clinical inference. Next, it analyzes the imaging study systematically by successively looking for more and more meaningful features that could potentially be anomalous until it locates a potential lesion. For this, it uses a combination of low-level image processing to highlight suspicious regions that are then classified as potential masses using deep learning networks, trained by prior anomalous data labeled by clinical experts. Watson then takes the clinical concepts derived from the imaging exam and case descriptions, and begins its reasoning process using clinical knowledge. Paths are explored and scored in the knowledge graph while searching for related concepts and facts that lead to the specified conclusions, starting from the chosen clinical concepts. The retained conclusion is the one which has overwhelming evidence from the number of high-scoring paths that lead up to it. The final clinical inference can be seen on the right. If you concur with Watson's inference, you can change your conclusion if you like. You can.
So, as I told you, finally it is left to us to take the result or accept the result or not. This is where the role of validation comes in. If you validate it, the system understands that it has done a good job. Please remember, the artificial intelligence also works based on the positive reaffirmation. If you pat its back, immediately it will understand, yes, I am doing a good job. Which means that in whichever instances it has done a good job, where you have accepted the result, it is going to keep increasing the accuracy. That is how the model will develop. That is why I told you in deep learning, it is a little more learning without any assistance. There is no need for supervision. It is a unsupervised learning with validation. That is where the deep learning will differentiate itself from the machine learning to predict accurately. To conclude this session, as I told you, the story is much more interesting, fascinating, and astonishing when I look up for the future. In future, what if I can develop? What if I can culture? Uh, I think I'll be old-fashioned if I use the term in future if I culture. In today's world, they are culturing the pluripotent nerve cell, neuron cell, pluripotent neuron cell line on a chip. Okay? Everything can be done aseptically. You can do the waste removal. You can do the input for the system. You can do controlled perfusion of oxygen, nutrients, and everything so that you can grow the cell mass on a chip. And externally, you can communicate from the chip to the chip, which means when you are communicating to the chip, you are not only communicating to the chip, you are communicating to the organoid which is developing on the chip. Whatever the organoid is going to understand from its environment, it is going to transfer it through the chip to you, and whatever you want to transfer as a model, as a information to the organoid, to a part of a brain, if it is becoming either externally or intrinsically in future, as Elon Musk is uh, putting a lot of investment on it, intrinsic organoids, it is very easy for you to understand how to get the uh, dialogue between a chip and a human being outside of the brain. This is really very fascinating, and this is going to be super useful in the brain disease management and treatment to understand the pathway in which it works and so on. So it is going to be super exciting. As good as any medicine, also, there could be side effects we need to understand. But if you approach it scientifically, if it is governed scientifically, if it is going to give only the positive results, absolutely, I am looking forward for these type of technologies to be in action along with you. So with this, I would like to conclude this session. Before I say before I take a few questions from you, I'm also equally excited to inform you that Biocon Academy has put together a new program in the area of artificial intelligence in life science, which is going to be a 4 months training program in association with IBAB, that is Institute for Bioinformatics and Applied Biotechnology in Electronic City Phase 1. They are extremely specialized in bioinformatics and artificial intelligence, machine learning, and deep learning. Biocon Academy is specialized in skill development in the biological sciences and life sciences. We are coming together to offer a program in artificial intelligence and life science to help a variety of students to get trained in this area and to help you get employed in this area. For more information about the career, for more information about the fees, for more information about the duration, curriculum, and so on, I really invite you to visit bioconacademy.com, go to the programs, select the students programs, and in the list you will see artificial intelligence and life science. It is called as SAILS, that is Syngene Biocon IBAB, Syngene IBAB certificate program in artificial intelligence in life science. I have to get used to this to say the whole full form. So I'm very excited to deliver this talk because it is very close to my heart and it is very interesting to talk about it, because most of the example what we have seen is not historic, neither it is futuristic. It is all happening today. That is the reason it is super exciting. I am more than happy to take few questions. Thank you very much.